All right, so we're going to start with focusing on the beginning of lab lesson number 3, which is talking about the cell cycle and mitosis. The first part of that activity is the forms of genetic information in the cell. So what I'm looking at right here is my model of the genetic information in the cell. When we're getting ready to do cell division, our genetic information is in this X shape that we see right here. So this X-shaped structure that my entire model represents here, so everything I see here, this is all called a chromosome. And we mentioned this last week when we were talking about our lab stuff. Remember that the only time that the DNA in your cell is shaped like an X, or it's condensed down into a chromosome, is when you're getting ready to do the process of division. So cell division, I have to split up my genetic information from each other. That's really easy to do when my information is all compacted down in this X shape that I see here. Normally, when we talk about the genetic information in a cell, it kind of is a big squiggly mess. Last week, I called it a spaghetti noodle mess. Normally, this is what your DNA looks like in the cell. So it's a mess all over the place. It's long and stringy everywhere. When we're going to do cell division, we package it down into an X, into a chromosome. Let me see in the chat, does anyone happen to remember what's the name of the loose form of DNA that kind of looks like a big mess? Spaghetti noodles, if you will. Do we happen to remember what that's called? So the ribosomes are an organelle. They're not a type of DNA, but that's a great guess. Yeah, Hannah's mentioning a word that's only slightly different from what we see on our list here, but it is important that it's different, called chromatin. Chromatin. My loose form of DNA that lives inside the nucleus of your cell is called chromatin. So think about it as a big spaghetti noodle mess. We've got this word right here, I'll draw your attention to it, called a chromatid. A chromatid is different. A chromatid is 1/2 of the X. So when I look at a chromosome, that's the X shape. Here, let's do a little bit of drawing here. My chromosome is shaped like an X. That's what a chromosome is. And I see that chromosome shape when my cell is getting ready to divide, so it can divide up its information. Each half of the chromosome is called a chromatid. So think about a chromatid as a half of that X structure that a chromosome is. So this over here on the left is a chromatid. This over here on the right is a chromatid as well. So a chromosome is a big X-shaped structure. It's made out of 2 chromatids. Those 2 chromatids meet in the center at a structure called the centromere. So the centromere is this central area that I see that attaches the 2 chromatids to each other. My centromere here in the middle. And the centromere is going to be important because when we do the process of mitosis, where we divide up the genetic information, we actually break this structure here so that I can split the 2 chromatids in half. So 2 chromatids attached at a centromere. Since we only have one label left, right, that tells me that this red stuff that I see here is the DNA. The DNA that we have inside of our cell, like we mentioned, normally looks like a chromatin spaghetti noodle mess. It's normally all over the place. But if you look at this picture, let's see if I can zoom in a little bit. If you look here, you can kind of see that the DNA that's inside a chromosome is twisted up in circles. So we have these special kinds of proteins that are called histone proteins. And you don't have to know those, but I'm just mentioning for you that the DNA is wrapped around histone proteins to give it this appearance that we see here, to make it very tightly wrapped up. There was a great question if the DNA is only on one side of the chromosome. No, the DNA is everywhere. So all of this pink stuff that we see is actually the DNA that's been all wrapped up. They've got the centromere made of proteins in the middle. This is just the part of the model where they show you that DNA is wrapped up. So technically, if we pointed anywhere on this pink stuff, all of that would be DNA. But the place that we labeled it on our model is right here. So this entire structure is made out of DNA, but when we talk about it in this form, in this X shape, we call this whole thing a chromosome. The chromosome is made of DNA that's arranged in these chromatids, these little half lines. Help me out in the chat. How do we feel about this so far, about the model? Thumbs up, thumbs down, or questions? Okay, Tierney says she's feeling good. Got some thumbs up. Awesome. Okay, so let me pull up, because I know you've got all those words. Let me pull up the fill in the blank words thing really fast, and we will talk about that. Okay, so I'm going to bear with me as we switch screens. Okay, so you should see now the text that you have in your lab packet. Remind me, is the text above or below the picture of the model? I can't remember how we arranged things this semester. The text is above. Okay, so this is the words, and it may not 100% match yours. I think we edited it a little bit this semester, but let's see if we can work together to fill in some of these blanks that you have in those words. So when we're looking at, let's put up a pointer here. When we're looking at the name of the genetic material in the cell, kind of the big picture term, what we call your genetic information, what do we call your genetic information in the cell? Does anyone know what we're getting at here? Yeah, so the broad term for the genetic information in your cell is your cell's DNA. So DNA is something we're going to be talking about a lot when we get into unit number 2. So lesson number 5, Marianne's asking what we're working on. This is the lab sheet. So lab sheet for lesson number 3. When we talk about your DNA, it's made out of a series of nucleotides. So A, T, G, and C. And you're going to learn specifically about those, like I said, in unit number 2. But everything that you are, your traits like your hair color, your skin color, your height, all of that is stored in information based on the order that we've arranged these little nucleotides in. So the genetic information in the cell, as you guys told me, that's DNA. It's made out of 4 nucleotides. When we talk about the loose form of DNA, this is the spaghetti noodle form of DNA that we mentioned before. What did we call that spaghetti noodle mess that I drew for you guys? Anybody write it down? This spaghetti noodle mess that I drew over there. The loose form, yeah. So the loose form is called chromatin. Chromatin. And remember, it's chromatin with an N. So chromatin is your loose form of DNA. Like it's mentioned in the textbook, you might read that it's called beads on a string. That's because it's got those histone proteins that we talked about. It wraps around them loosely. But when it's time for a cell to divide, it's going to be really hard to divide that spaghetti noodle mess equally between 2 cells. So what we're going to do to divide it equally between 2 cells is build those X-shaped structures. Those X-shaped structures are what that entire model showed me. What was the name again of that big model, the big X in the model? Yeah, the form that your genetic information, when you're dividing, is in is a chromosome. Chromosome. So the big overall X, what we see inside our cell, that's called a chromosome. Remember that each half of the chromosome is made of things that are called chromatids. Chromatids. So here's my wink, wink, nudge, nudge. Make sure we don't mix up a chromatid and chromatin, 2 very different things there. So we've got 2 chromatids that are held together in the middle with that middle structure we labeled. called a centromere. Centromere because I'm in the center of a chromosome, my center. And remember, the job of that centromere, there's two jobs. Job number one is to hold the chromatids together. Job number two is to help out with the process of mitosis. Now, here's something that I want you guys to write down somewhere on your lab packet or on another piece of paper that you're not going to lose. When we talk about mitosis, we're talking about specifically the division of DNA. Mitosis is the division of DNA. I don't like this terminology here. Mitosis is the process that a cell uses to split up its genetic information. I actually have a different process that I use to split up all of my organelles, to split up the soupy stuff in my cell. That other process is called cytokinesis. Cytokinesis. That's the name of the process that divides up the soup that's in the cell, divides up the thing called the cytoplasm. In mitosis, all that we're splitting between the two cells is their genetic information. So we're taking those X-shaped chromosomes, we're going to split them in half and send them to different places so that we can divide these cells. So we use a special set of proteins to do that. Those proteins are called the mitotic spindle. The mitotic spindle. And we will see the mitotic spindle when we start looking at pictures of the process of mitosis. On my pretty stained pictures that show lots of cool colors, they're going to be big, long green proteins. And their job essentially is to play tug of war with the chromosomes. So we pull it toward one side, then we pull it toward the other side, and we pull back and forth until we actually split when we divide them up. These tug of war ropes that we're using to pull the chromosomes back and forth, they're actually attached, lost my pointer there, they're actually attached at the ends to one of our organelles from last week. The organelles called the centrioles. And if you remember from lab last week, we said they kind of look like churros or licorice. Those are my teams in the big tug of war with my chromosomes. My centriole on one side pulls the chromosome one way, and then my other centriole pulls it the other way, and they pull back and forth until those chromosomes split. And when they split, they end up in two different locations in the dividing cell, which is ultimately going to allow us to make two new cells. I'm pretty sure you have a word or have a blank in your packet somewhere that I don't have in this list of information. Was it cytokinesis? That was that other blank that we had, or was there more than one that I missed? No, okay. So hopefully that helps you with some of the terminology of the process of mitosis. We will use a lot of these terms as we start going through and looking at the phases of mitosis. So hopefully this brief definition of them or description of what happens helps you out as we start using those terms more specifically. So let me go back to the PowerPoint I made for today. You have access to this image in this week's guided lesson. So you don't see it on your worksheet, but it is in the week's guided lesson. What this shows you is the whole process of the cell cycle. So we're going to spend a lot of time in the next couple of weeks looking at different types of cells. In all of your cells in the body, though, they have a very predictable way that their life goes, kind of like we as humans have a predictable way that our life goes. So we start, the beginning of the cell cycle is as soon as a cell is formed. When a cell is first formed, it's got to go through a whole bunch of growth. So the first thing a cell does when it's made, just like a baby, is it's got to grow big and strong. So cells go through this big phase of just growing, making more proteins, building more energy. They do a whole lot of stuff there. Then they kind of reach maturity where they realize, oh, maybe I would like to divide. Maybe I would like to become 2 cells instead of just 1. To get ready to become a 2 cells instead of 1, to do the process of division, the cell has to copy its DNA. So there's this portion of its life cycle that's called the S phase. The S phase is when the cell is not only growing, but it's also synthesizing new DNA. Once it's copied its DNA, it goes through one more major growth phase to get it ready to do division. So most of a cell's lifetime, as you can see from the size of these phases on my wheel here, most of a cell's lifespan is spent not dividing. And there's a term, you can see it on the image here. The parts of a cell's life cycle that they're not dividing is called interphase. Interphase. Interphase is not a stage of mitosis. Interphase is a part of the cell cycle where the cell is growing and where it's copying its genetic information. Once it's grown big enough, once it's copied its genetic information, then it's ready to enter the process of mitosis. So mitosis, I guess I have it labeled up here, mitosis includes 4 specific phases, where there's 4 different things that a cell does to do the process of mitosis. And this would be one of those things that we just need to memorize in order. The easiest way to memorize the phases of mitosis is just to memorize PMAT. PMAT, that's the order that they go in. We start with prophase. That's the first phase when a cell has decided it's going to divide. It does prophase. Then it transitions into metaphase, where its chromosomes meet in the middle of the cell. Then we go to anaphase, where my DNA actually splits, my chromosomes break in half and start traveling away from each other, into telophase, the last phase, where we pull apart the 2 cells from each other. Remember that the process of mitosis only has to do with genetic information. This is all about the DNA. Because here's that phase that I mentioned to you, or that process I mentioned to you before, called cytokinesis. Cytokinesis is when we divide the cytoplasm. So cytokinesis is when we divide the cytoplasm. Notice that the process of cytokinesis, it's not part of mitosis. It happens mostly after mitosis is done, but you can see this little creeping line right here. Cytokinesis actually happens a lot during the mitosis phase of telophase, and that's going to be the way that we actually most easily identify telophase, as we start to see the process of cytokinesis happening as well. So I had a question in the chat. Do you have this picture? Again, you have this picture in this week's guided lesson for lab that shows you the phases of the cell cycle. So check out that guided lesson. You can access it through Visible Body. There's a link to get there that has a picture of the cell cycle for you. So here's an important idea for us to write down. The cell cycle has 3 phases, or 3, we'll call them parts actually, 3 parts of the cell cycle. The first part of the cell cycle is interphase. Interphase is when a cell is growing and it's copying DNA. That's the first part of the cell cycle. The second part of the cell cycle is mitosis, when the cell divides its DNA. That's the second part. And the final part of the cell cycle is called cytokinesis. Cytokinesis, when we divide the cytoplasm. This would maybe be I call it an underline highlight star idea. This is important. So knowing that first a cell grows and copies its genetic information, then it divides that genetic information, and it divides up its soupy stuff and its organelles. That's the 3 phases, or the 3 parts of a cell's life cycle. Now, we are primarily focusing in our lab class on mitosis. on this small period of time when a cell is going through and dividing from one cell into two. So I'm going to transition into, again, looking at some pictures from that guided lesson that show you what the process of mitosis looks like. Keep in mind that for most of a cell's lifetime, it's not dividing. Most of the time, it's not doing mitosis. So here's that first part of the pictures. Again, in the guided lesson, you've got these. So first thing we'll mention on our picture here, interphase. Remember, interphase is not part of mitosis, so the cell is not dividing. All the cell is doing right now when it's in interphase is it's growing and it's copying its genetic information. Now, I'm going to go through and talk about each of the phases with you, and I'm going to describe for you how you would identify them. You should definitely take some notes about the specific things that you see when a cell is in these phases, because in just a little bit, we're going to pull up some slide pictures that show cells, and I'm going to ask you to identify what phase they're in. So I'm going to give you some helpful hints to identify the different phases of mitosis on a slide, and then we'll practice identifying them. So when we start here with interphase, remember, interphase is not part of mitosis, but interphase is what normally a cell is going to be doing. It's going to be living most of its life in interphase. When a cell is doing its normal life, when it's living in interphase, notice that we can very clearly see a nucleus, and inside that nucleus is our chromatin. So remember, chromatin is that spaghetti noodle mess that we have of the genetic information floating around here inside the nucleus, chromatin. So if you see, let's go back up to our image. Our cells, unfortunately, are not nearly as beautiful as these. Ours aren't colored, but the genetic information in these pictures is blue. Notice how we just kind of see a fuzzy blue appearance here. My nucleus is completely filled with genetic information. If there's no kind of checks like you see in here where there's like polka dots in this nucleus, this is how we know that we have chromatin and not chromosomes. When I start forming chromosomes, you start to see little polka dots in the nucleus because that's taking your spaghetti noodles and putting them into little X's. So when a cell is in interphase and you're looking at it, you're going to see a nucleus that on our cells is pretty much purple everywhere. There's no polka dots, there's nothing in there except a dark purple nucleus. That's a cell in interphase because its genetic information is still chromatin. Once we start the process of division, our first phase is prophase. And there are two different ways that prophase can look on a slide. So I'm going to describe for you what the two look like, but you will never have to distinguish between early prophase compared to late prophase. That's not going to be something that you have to do. If we're talking about early prophase, so the beginning of the process of DNA division, that's when we start to see those polka dots in the nucleus of the cell, because those polka dots are the chromosomes that are condensing down into lines of genetic information instead of just a squiggly mess. So the first way you might see a cell in prophase is if you look at its nucleus and it looks like we still have this solid nucleus, but there are dots inside of it. That's a cell in prophase. That's a cell getting ready to start dividing its genetic information. By the end of prophase, what has happened is all of that genetic information has formed into chromosomes. So each of these little blue things that you see here, all of those are chromosomes. But at the end of prophase, here's a note we should make for ourselves. At the end of prophase, there's no longer a nuclear envelope. We no longer have that membrane on the outside of the nucleus. So see how these chromosomes are kind of all over the place in a mess? That's because the nuclear envelope has dissolved and it's not there anymore, but my chromosomes haven't really started moving yet. They're just kind of loose, floating around in the cell. That's what late prophase looks like. Looks like chromosomes that have no particular order to them floating everywhere. After prophase finishes, we transition into what's called metaphase. And the way you're going to identify metaphase is a line of chromosomes in the middle of the cell That line of chromosomes in the middle of the cell is called the metaphase plate. So the metaphase plate is when all of the chromosomes meet in the middle of a cell. And the reason that all of those chromosomes meet in the middle of the cell is because my centromere, or excuse me, my centriole on this side starts pulling on the chromosomes, and my centriole on this side starts pulling on the chromosomes. And these two centrioles are very equally matched. They both pull with the same amount of strength. So all of those chromosomes end up lined up in the middle of the cell because this side tugs a little, then this side tugs. They pull back and forth, but we end up with all the chromosomes in the middle of the cell. So metaphase is probably the easiest phase of mitosis to identify because all of those chromosomes are in the middle of the cell. They're all meeting, metaphase. Eventually though, when I pull on those cells long enough, or those chromosomes long enough, they end up splitting from each other because the tension is just too strong. So here's my metaphase right here. We're all lined up here in the middle. If I pull and pull and pull on these, what will happen is these X's will split in half and start traveling to opposite sides of the cell. So you'll see a group of genetic information that's traveling toward this centriole, excuse me. I didn't have coffee this morning, full disclosure, so I'm feeling it. I've got half of my genetic information traveling down toward this direction, half of my genetic information traveling up this direction. So see how those X-shaped chromosomes that I had right here and my metaphase plate, they've split into little V's that I see down here. So I've got half of my V's that are traveling up to this side, half of my V's that are traveling down to this side. This is also another phase that would be pretty easy to recognize because we have two different clusters of genetic information. So one of them up top, one of them down below. That's because they're traveling in two different directions. They're going to end up in two different cells. The last thing that happens in the process of mitosis is telophase. And telophase is the final phase of mitosis. A couple of things happen in telophase. The first thing that happens in telophase is we start to make a new nuclear envelope. So we start to form what will be the nucleus in the new cell we've just created. So you can see on my sketch down here, they've got a little line where we're starting to form that nucleus. But because we're doing division of the cells, we're actually making two new nuclei. So the first thing that happens in telophase is we make a new nuclear envelope to cover up our genetic information. The second thing that happens in telophase is those chromosomes that we had so tightly packed up, they'll actually start to loosen again. They're going to go back to being chromatin, be a big mess that I see inside the nucleus. So when you're trying to identify telophase, you'll see two clusters of genetic information that are very far separated from each other. They're completely divided. Those two clusters might be fuzzy looking instead of having little strings in them if we've started to take those chromosomes and decondense them, so make them back into the chromatin. The other way you can identify telophase though, and it's kind of cheating, but it works, so we're going to go with it. You can also look for the little indentation. See how I have an indentation right here and I have an indentation right here? You can see it down here on the sketch as well. We have those indentations on the two sides. Those things are called the cleavage furrow, and it's labeled right there. I'll circle it, the cleavage furrow. The cleavage furrow is when the plasma membrane of the cell starts to fold in on itself. Because if we're going to split these cells in half, we literally have to pinch them in the middle, squeeze them back down together. So you start to see a cleavage furrow, an indentation between the cells, technically squeezing together the plasma membrane and dividing up the cytoplasm. Technically, that's all stuff that happens in cytokinesis. Remember, cytokinesis is not part of mitosis, but it happens at the same time. So technically, cytokinesis is what we see when we form that cleavage furrow, but cytokinesis happens at the same time as telophase, which is why I can use it to help me identify a cell that's in telophase. If I start to see that indentation, that cleavage furrow, then I know we're doing the process of telophase for the DNA, or we're doing the process of cytokinesis if we want to know what's happening with the cell. I've talked way too much. I need to pause for a moment. Help me out in the chat. What questions do we still have? Or what phases do we want to review again? Help me know how to help you. Hannah's got a question. Go for it.
Okay, so I just wanted to like clarify and everything real quick. So cytokinesis and telophase happen at the same time and everything, and then cytokinesis is different from mitosis. But even with the labeling, with the two cells coming together or like separating, this is going to be a really weird question, but in some instances, would we use telophase and cytokinesis in the same answer for a question if it was like labeling a cell that looks like that's separating? Could we use both answers, or would we be safe using one or the other? I don't know if that makes any sense.
Yeah, no, that's an excellent question, and I'm glad that you asked that, because it gives me a chance to help you know how to tell which one you should be using. So great question. The way that you're going to know whether you're supposed to tell me telophase or cytokinesis is based on the way the question is asked. So let me type the two ways we could ask the question. The first way we could ask the question would be which, I can't remember, I think which part of the cell cycle is this cell in? That'd be the first way I could ask that question. The other way I could ask the question is which phase of mitosis is this cell in? If you have a question that asks you to identify the phase of mitosis that this cell is in, the phase of mitosis, or the step in the process of cell division, of DNA division that this cell is in, this is in telophase, because that's a phase of mitosis. If I ask you about which part of the cell cycle this cell is in, the parts of the cell cycle that we'd be asking for on the question are either cytokinesis or interphase. So your tip off with, do I say telophase or do I say cytokinesis? Is it asking for the part of the cell cycle, or is it asking for the phase of mitosis? And I think you have a little box on your worksheet that mentions that. That would be an underline highlight star. Make sure we catch that. Because yes, we do see both things happening in this picture. And the way that you identify telophase, the phase of mitosis called telophase, is by looking for that cleavage furrow forming, is by looking for the process of cytokinesis happening. But if I'm asking you about what phase of mitosis is going on, this isn't part of mitosis. So technically we would say that this is telophase. And on the flip side, right, if I ask you which part of the cell cycle we're seeing here, the specific part of the cell cycle we're seeing is the process of cytokinesis, dividing up that genetic information. That was a great question. Thank you for asking that question, Hannah. Yeah. I also have a question on the slides. Are we looking for let me scroll down, a specific part of prophase or just prophase in general? You're just looking for prophase in general. We're never going to ask you to specify, is this early prophase or late prophase? But along those lines, you do need to be able to identify prophase with either of them. So you'll have to be able to determine that a cell that's in early prophase is in prophase, and a cell that's in late prophase is also in prophase. So make sure you can identify both of them. We're not a stickler on the homework or on the tests for whether it's early or late. Just know that it looks a little bit different in those two phases. Yeah, another great question. Any other questions before we start looking at slides? Okay, well, let me pop up for you. I made this slide last night, so you probably don't have it anywhere if you want to snap a picture on your phone. I know you have examples in the guided lesson that show you the same things, but this is something that I built last night. This is showing us the steps of mitosis in a circle. So remember, we do need to know what order they go in. We start with prophase, then we go into metaphase, then we go into anaphase, and finally telophase. So remember, PMAT is the order that we go in here. So remember, in the process of prophase, some of the things that happen is the nucleus dissolves. We also see the chromosomes start to form. This particular cell is a cell in prophase because see all of these little spots, these little dark things I see inside here? Notice, like I mentioned, our cells aren't beautifully colored like those pictures I showed you. So here's my outer membrane, the plasma membrane of this cell. Inside the cell, we've got a lot of proteins, a lot of other stuff. My genetic information is the darker colored stuff in here. It was blue on those slides we looked at before. So my genetic information is dark pink or maybe purple here. So I've got spotty purple genetic information inside here. I also don't have a distinct circle around this group of genetic information. So my nuclear envelope has dissolved and I've started to form those chromosomes. Both of those things are what happens in prophase. This particular cell is a cell in the process of prophase. Remember, I told you that metaphase is the phase that's probably going to be the easiest for you to identify because in metaphase, the chromosomes meet in the middle of the cell. So see this line of chromosomes here where they're meeting in the middle of the cell? This whole thing, by the way, all of this is a cell. So inside that cell, now I've got that line of chromosomes. You can kind of see those green proteins from my previous pictures that they're going from those centrioles, attaching out my mitotic spindle that's pulling on these chromosomes here in the middle. So here I am in metaphase. I've got a line of chromosomes in the middle of a cell. Then I transition over here into anaphase. In anaphase, I see two different sections of my genetic information. It's being pulled toward a centriole right here, and it's being pulled toward a centriole right here. The difference between anaphase, where my chromosomes are being pulled to opposite sides, and telophase comes down to that thing we were just talking about, right? That cleavage furrow. So see this line, this divot right here, and this divot right here? This cell is pinching in on itself. So its plasma membrane is squeezing down from both sides to squeeze it off, to attach it, or to detach it from each other. So I am in the process of telophase now. My genetic information is completely divided. I know that because cytokinesis is happening. It won't happen until our genetic information is divided. Also check out this area right here. See how my genetic information up here is kind of fuzzy looking? This is genetic information that's starting to decondense. This is information that's loosening back up to be chromatin. This side still looks like chromosomes, so we still have this one condensed, but this side is starting to loosen up. Because it's loosening up, I know that I'm in telophase, because that's what happens in telophase. We decondense that genetic information again. So the 4 phases of mitosis. Phase, where I can see the starts of forming chromosomes in there. Metaphase, where those chromosomes are lined up in the middle, where they meet in the middle. Anaphase, where they've separated from one another and they're being pulled away from each other. And telophase, where they're completely separate and start to decondense. Also, we can see that process of cytokinesis happening as well. Those are our 4 phases of mitosis. In the chat, give me a thumbs up, give me a thumbs down. How do we feel with the pictures labeled? Let's start with the labeled pictures. Okay, I'm getting some thumbs up, I'm getting some smiles. Perfect. Okay, so here's what I'm going to do. I am going to split you guys into groups. Oh, Nicole's got a hint for us. If you want to print the slide, hide the chat box. Let me go and see it. Hide the chat box and right click to print in landscape. Awesome. Yeah, that's a great idea. So hide the chat box and then right click on your screen, and that will allow you to print. That's a great pro tip. Thank you, Nicole, for that. So I'll give you a couple of seconds to print your screenshots really fast. And then what we're going to do is I'm going to split you guys into groups. I'm going to show you the picture of what you're going to work on with your groups, because then, as you guys know, I have to go in and add it to your group so you can pull it up. So let me show you the picture that you're going to be working on with your classmates. If we were in lab together and we had our microscopes out, we would see something like this when we were looking at a slide. Lots of different cells all over the place in lots of different phases of mitosis. You and your group are going to work on identifying those phases. So if you want to snap a picture of this on your phone, it's going to take me a moment to get these loaded into your groups. But at this point, I'm going to go ahead and split you guys up and give you about 4 or 5 minutes to talk about these cells, and then we'll come back together. So let me get those groups started, and I will upload this file for you. All right, welcome back from our groups. How'd we do? Were we able to name our phases?
C.
Oops, sorry. Turn your mic back on. I accidentally bumped you off. Go for it.
You're fine. C and B looks kind of similar. Like I knew A and D for sure, but like B and C I was kind of stuck on personally.
Okay, so let's chat this out as a group and see what we thought. Well, so let's start with this. We mentioned that we felt pretty good about A and C. So somebody from group number 1 and somebody from group number 2, if you can click on that T above your screen there, that'll allow you to type. Do me a favor and let's fill it in. So group number 1, type your answer pretty far to the left. Group number 2, try to type it close to the right so we don't type over each other. What phase of mitosis did we think that A was in? And what phase did we think that C was in? Or D, sorry, it was B, or I don't know what it was. A and D, I think those were the ones that we felt good on. Okay, so one of my groups typed in. Perfect. Okay, so it looks like we are agreeing. D is definitely an example of metaphase, absolutely, because we can see those chromosomes meeting in the middle of the cell. Perfect. And one of my groups voted anaphase for A, and I would agree with that. I'll mention for you a couple of reasons that I think this is anaphase. The first one, see how all of my genetic information seems to be angling toward the middle of the cell, so it's here in the middle. That would mean for us that this genetic information is still moving. When we stop pulling this genetic information this direction in the cell, all of these little strings will just kind of float in the middle of this space. Same thing with these ones here. They would just start kind of floating, pointing in different directions. So these are still moving. In the process of anaphase, my 2 sets of genetic information move away from each other. So first reason I think this is anaphase is because that genetic information is all stringing toward the middle. Second reason I think this is anaphase and not telophase is we don't have a cleavage furrow, right? We can't see a place where those cells are where the cell is starting to divide its cytoplasm as well. So this would be another cell that we had in anaphase here, another reason that we saw that. Okay, so we feel good about our metaphase. We feel good about our anaphase. So help me out. Let's type it over here. If there was more than one phase that we thought these cells could be, can you guys type for me what we were thinking B and C were? What were some of the options that we thought for B and C? Or maybe the comment was just that they were similar. If we thought they were similar, I did have that note for your groups that it could be the same phase. Yeah, and our groups, it seems like, kind of agreed. These are our two examples of prophase for us. So you can see that genetic information starting to clump up inside here. So we're starting to see chromosomes form. Same thing here, I'm starting to see those chromosomes form. This one, what I liked about this one is you can still definitely see the nucleus around the genetic information, see that purple line. Whereas this one, it looks like the nucleus is starting to fall apart because I have some spaces where I don't really see that nuclear envelope anymore. So both of these are an example of cells that are in prophase. Excuse my 3-year-old, so sorry. Both of them are cells that are in prophase, but this one we would probably say is late prophase. And for that, if you sneak a peek over here, see this genetic information right here, and there's no envelope around that at all. So that's definitely late prophase. So these two are probably late prophase. This would probably be an early prophase right here, because I can see the chromosomes, but still that envelope. Same deal with this one right here. See how you can still see that envelope around it. In terms of cells that are in interphase, which is not part of mitosis, right? But cells that are in interphase, we said that their genetic information isn't really clumped up at all. We can't see it very well. Some of these cells, how when you look at them, you don't even really see the nucleus in them. Some of these cells would be interphase cells because I can't really see their genetic information. It's really stringy and it's just hard to see. So some of these would be our interphase cells that aren't even doing mitosis. Once I start clumping up that genetic information, you start to see it, and that's when I'm in prophase. And we got metaphase here, we got anaphase here. I don't really have a telophase on this slide. Let me see. Bear with me here as I bounce. Let me see if I can find you a good example of telophase, because I know that we've got some good ones. Let's see. Let's just do some looking together. Actually, we'll look at good and bad together to practice this. These pictures that I'm showing you, you guys have access to them. There is a link in our Blackboard site that says the Student Google Drive. The Student Google Drive is a place where you could go to download your worksheets, but it's also a place that has a ton of tissue images. So look for that link to the Student Google Drive. There is a folder in there that says tissue images from our slides, and this is just the PowerPoint called Mitosis. So you have access to this that you can pull this up to study from later. When I'm looking at this picture here, there's obviously a super great cell right here. Check out this one. What phase do we think we'd say this one right here is in? If we were to guess, what phase of mitosis do we have going on right here in the middle? Yeah, a couple of us are voting in anaphase. That's correct. Remember, we can see those chromosomes that are splitting apart from each other. They're still dragging toward the middle that direction. So this is a really good example of a cell that's in anaphase. Up over here, notice how this cell has some clumpy DNA inside of it. This would be a cell that we would say is in prophase. This one over here, I would probably say is an interphase cell because that genetic information isn't distinctly clumped like I see over here. but it's all kind of fuzzy in an area together. So that could be potentially a prophase or an interphase cell, excuse me. Not my favorite. Also not my favorite. I know we have a really awesome telophase in here. Let me find it. Okay, check this one out. This may have been the one I used before even, I don't know. I probably was. So we have these two distinct nuclei where I have genetic information, and we can see that cleavage furrow. So here's another example for us of the process of telophase. Here's another telophase. See how I can see the cleavage furrow here on the top and on the bottom, and how I've got these two dark circles of genetic information. They're not moving away from each other anymore like we saw on some of those other slides. They're already moved. They found their new locations. Now we're just dividing up the cytoplasm to get these cells completely separate. So there's some telophase for us right there with those, again, with those cleavage furrows that we see in the middle. I think this is actually the picture that you guys did with your group. So remember we said these were the anaphase cell, because the genetic information is being pulled away from each other. And there's that metaphase cell with the line of chromosomes in the middle, and then a whole lot of prophase cells around it where that genetic information is clumped. The question came up again where these slides are. So when you're in the lab folder, so lab resources area of our Blackboard site, so go to lab resources. There's a link that says it's to the student Google Drive. The student Google Drive, as long as you're not logged into your personal Gmail account, when you click on that link, it's going to take you to an online drive that has a whole bunch of resources in it, including a folder that says tissue images from our slides. Let me type it. Tissue images from our slides. And this particular set of images is called mitosis. So I'm not going to go through, I think that's probably all I'm going to hit here. But definitely take some time to practice. This is a pretty good one. Tell me what phases we see on this slide. What phases do we see on this slide here? Yep, so someone's saying we got metaphase. Look at that awesome metaphase up here. I've got an anaphase over here. Hey, I've got another metaphase right here. Yeah, and someone's mentioning a prophase. There's a prophase with no nucleus, just all those little chromas down there. So, yep, we got a prophase here. So this one has a bunch of the phases on it as well. Here's another cell. This image is a little bit washed out, but here's another one that we would say is in telophase. No, this one is pretty much completely divided from its neighbor. So we can see where it pinched off in the very middle. And we have two little nuclei with the genetic information that's right there. So this cell, if we were to talk about a late telophase, that would be this cell right here. We can see its genetic information in two separate places. And that genetic information is decondensed. It's fuzzy looking. And we can see that cleavage furrow very clearly. So there would be a telophase cell. Here's another prophase right here, because I can see the little polka dots, and another little anaphase over here. Phases of mitosis: thumbs up, thumbs down. What do we think? How do we feel about mitosis phases? Or what questions do we have? Hannah says she feels better. That's awesome. Better is where we want to be. It just takes practice. We're going to have to keep practicing looking at stuff. A question, Jacqueline? Or somebody has a mic on? Yeah, did you, or maybe it was on accident. No worries. Okay, so I've got about 10-15 minutes left. Here are the options. The options are either we can talk about the mitosis models, or we can talk about the stuff at the beginning of lesson number 4 that has to do with the 4 types of tissue in the body and how we name epithelial tissues. Let me put the vote to the class. Do we want to do the lesson 4 stuff, or do we want to do the mitosis models? I got 2 votes for lesson 4. Anyone else want to vote? Lesson 3 or lesson 4? Okay, well, it appears that I have an overwhelming voting majority that would like to do lesson number 4. So we'll go ahead and do that. And if you have questions about those mitosis models, maybe email me about them. And the guided lesson has labeled pictures for you. So I think that'll really help with that as well. Lesson number 4 is going to take us a few weeks actually to finish because it covers a lot of different content. So this is the beginning, very close to the beginning of lesson number 4. You have 4 pictures of the tissue types in the body. So we're going to talk about which kind of tissue we see in each of these images, and I'm going to give you an idea of some of their major characteristics. Part of your assignment this week is you'll see some pictures of tissues that we have on our slides from the class, and we'll ask you which of the 4 main types of tissue they are. So I'm going to give you some characteristics or some things to look for to help you know which types of tissue look like what. This image that I see right here, this is a picture of the type of tissue that we call epithelial tissue. Epithelial tissue. When you think of epithelial tissue, the easiest way to remember where you'd find it, this kind of tissue is called covering and lining. Lining tissue. As in, it covers the outside of your body and it lines the inside of your body. I haven't cracked this joke very much this semester because we haven't had a lot of time together, but I would say that technically my favorite organ system in the body is the digestive system, because I did my PhD work studying how the bacteria that lives inside your digestive system can actually influence the way that you think and feel. So that's my fun little side project that I'm interested in. So I like to joke that my favorite system is lined with epithelial tissue. So from your mouth all the way through your anus, you've got epithelial tissue that lines that part. You see epithelial tissue lining your thoracic cavity or your abdominal cavity. But epithelial tissue is also what covers the outside of your body. Some characteristics of epithelial tissue. So all this space right here that I see, all of this is epithelial tissue. And epithelial tissue is pretty much 100% cells. So it's made entirely of cells. Those cells can either be like we see in this picture where they're stacked on top of each other, or it can be that we just have 1 or 2 layers of those cells. So epithelial tissue, known as the covering and lining tissue, all it has in it is cells, lots and lots of cells. Another type of tissue that is pretty much entirely made out of cells is this image that we see down here. This is an image of muscle tissue. Muscle tissue. Now, muscle tissue is made out of cells. These cells are, in the case at least of this type of muscle tissue, are long and cylindrical cells. But the big thing for you to know about the cells in muscle tissue is that the cells are filled with proteins. Filled with proteins. And these are proteins that we're going to spend a lot of time talking about toward the end of the semester. A lot of them are contractile proteins. So allowing you to move your muscles, allowing you to push food through the digestive tract, allowing you to pump blood through your body. Contractile proteins help muscle tissue do contractions. So muscle tissue, the main type of muscle tissue that we'll see a lot of is skeletal muscle tissue. That's these big, long cells. But we will also see what's called cardiac muscle tissue that's inside the heart, and we'll also see some smooth muscle tissue. Muscle tissue, another type of tissue with lots and lots of cells. The type of tissue that we're looking at over here is one example of what we call connective tissue. Connective tissue. Now, when you are identifying tissues in the body or when you're looking at slides, if you don't know which of the four main types of tissue you're looking at, you might be safe to just assume it's some kind of connective tissue, because connective tissue is the most diverse kind of tissue in the body. So we have connective tissue includes things, for example, like your bone, but it also includes things like blood or adipose tissue, or the kind of tissue we're looking at in this picture right here is called areolar connective tissue. So connective tissue has the most variety. The big thing with connective tissue is that there are cells plus something called extracellular matrix. Extracellular matrix. Now, I know we've used this word before, extracellular. Can you help me out in the chat? What does extracellular mean? Extracellular was a lecture word. No, we're doing lab today. Yeah, exactly. Extracellular means it's outside the cell. So connective tissue, the big way you're going to identify it is you will see cells inside of it, and then you're going to see a bunch of stuff on the outside of the cells. So some of that stuff on the outside of cells, a lot of different proteins. But you might also see stuff, for example, outside of bone, we have calcium. Outside of my bone cells, there's calcium. Outside of, we're going to talk about cartilage. So outside of my cartilage cells, we have something called chondroitin sulfate. Chondroitin sulfate. You'll know that later. You don't have to memorize that now. But we'll see these cells that have a mixture of stuff on the outside. So in this picture, each of these little dots that I see right here are my types of cells. Those particular cells are called fibroblasts. Fibroblasts. Its name tells me what it does. It is a fiber builder, fibroblast. So I can see in my picture here my fiber builders, my fibroblasts, and then see I've got these dark protein fibers, I've got these thick pink protein fibers, I've got some really skinny protein fibers. The big idea with connective tissue is if you see a bunch of different proteins on the outside, or if you see a bunch of stuff on the outside of the cells, that's connective tissue. It's got a bunch of stuff outside those cells. And then finally, we're looking at my last example down here is nervous tissue. And nervous tissue has two types of cells in it. The first type of cells, which are the big ones, are the neurons. I like to call those the thinking cells because they're literally the ones that help you think. But the nervous tissue also has what are called neuroglia. And the neuroglia are these little tiny dots, these little cells that we see around here. Their name literally means that they're neuron helpers. And that's the job of these cells, is to help my neurons do the thinking or the processing. So, four main types of tissue in the body. Epithelial tissue, made entirely of cells. Connective tissue, made of cells plus stuff on the outside. Muscle tissue, cells with lots of proteins inside of them. And then nervous tissue, with these weird shaped neuron cells and their little tiny neuroglia, they're helpers on the outside. That's the four main tissue types. Next week, we are going to be focusing in lab a lot on epithelial tissue and also on connective tissue. So when we're talking about epithelial tissue, there are two parts to its name. The two parts to the name of epithelial tissue is the shape of the cells, as well as the number of layers of cells. So what I mean by that is when we look at these 3 different examples of what shapes of the cells could be, these cells look a little bit like cubes. For my friends who had a chance to do this part of the lesson, for cells that are shaped like cubes, what do I call this shape of an epithelial cell? Not a trick question. Yeah, Nicole chimed in. I gave you all the hints, right? This shape of epithelial cells, these are called cuboidal cells. Cuboidal cells, as in I'm shaped like cubes. Cuboidal cell. The ones over here on the right are called columnar cells. Columnar, as in I'm shaped like columns, or I'm a lot taller than I am wide. With a cuboidal cell, it's just as wide as it is tall. With a columnar cell, it's a lot taller than it is wide. That's columnar. This last shape of cells is really flat and squished or squashed, we might say. These ones are called squamous cells, or sometimes people call them squamous. You can say it either way, don't care. Squamous cells or squamous cells, these are the squished cells. So they're going to be really flat, maybe a little bit circular if you're looking at them from the top, but we're flat. So the squished flat cells, that's my squamous cells that I see down here. My just as wide as they are tall cells, that's the cuboidal cells. And my much taller than I am wide are called columnar cells. Each of these examples show one layer of cells. In epithelial tissue, the word I use for one layer of cells is called simple. So simple epithelium has one layer of cells. Stratified epithelium has at least 2 and a lot of times many more layers of cells. So when we talk about epithelial tissue, its name is going to tell me what the shape is of the cells that are inside of it, and it's going to tell me the number of layers that it has. So here's a picture that you have in the guided lesson right here that tells you the different types or shows you the different types of epithelium. Notice how on my top line up here, all of these are simple epithelium. Simple means there's one layer of cells. Stratified means there's more than one layer of cells. There's this weird word right here, pseudo stratified. Pseudo means fake. So pseudo stratified epithelium means that we have fake more than one layer. When you look at it, it's pretending that it has more than one layer, but there's actually only one layer of cells. The way you would tell that cells are arranged in a layer, by the way, is by looking at their nucleus. So see these purple dots that I can see right here? These purple dots that I see right here are the nuclei of my cells. So when I see them arranged in one line and only one line, that tells me it's a simple epithelium. When I see them arranged in 2 lines, for example here, or I see lots of different lines of cells here, that's a stratified epithelium. See how with pseudo-stratified columnar, the nuclei are all over the place? That's actually the way that you identify this type of tissue. If those nuclei aren't in one line, but they're also not in 2 lines, it's probably pseudo-stratified. One last type of epithelial tissue that's name does tell us about its shape, but it makes us kind of sad on the inside. This is called transitional epithelium. Transitional epithelium means that its shape changes or it transitions. So this type of epithelium is going to have lots of different shapes of the cells. There's going to be multiple layers, but the big way we'll identify it is that we have this type of epithelial tissue that changes shape. So transitional epithelium changes shape. Simple epithelium, one layer. Stratified epithelium, more than one layer. And my fake it till you make it, pseudo-stratified epithelium. I am about out of time, so I want to open the chat for any questions that you have to make sure I I address those in particular since we're about out of time. Do we have any particular questions about our tissues or our epithelium or all that jazz? Or give me a thumbs up if we feel good. Nicole asked, how far in lesson number 4 do we need to go? It's just the first 2 pages. So beyond the naming the types of epithelium, there's also a couple of notes in there. I'll bump to a slide just to show you some pictures. Again, from your guided lesson. There's also some notes about things called cilia, which look a lot like eyelashes, versus microvilli that kind of look like peach fuzz on top of our cells. So the last thing that you're doing in lesson number 4 is when you are making predictions. So if I need to do a lot of absorption, or if I need to move something in and out of parts of the body, or if it's an area that stuff needs to travel across, you want to make some predictions based on what we talked about with the simple versus stratified, or what you'll read about these cilia versus microvilli. So bottom of page 2, that's as far as you have to go for this week's lab. Good question. Any other questions? Again, I apologize for my daughter. 3 is a hard age. All right, well, if we don't have any other particular questions, I'm going to go ahead and end the recording and turn off the mic, but I'll stick around if you want to type any others in the chat. Otherwise, have a great afternoon. I will be back tomorrow morning at 9:30 for us to cover the first half of lecture lesson number 3. So make sure you're working on that today to get you ready for that session tomorrow and the session on Thursday. So goodbye to my friends on the recording.